अस्सलाम वालेकुम स्टूडेंट्स, होप यू ऑल आर फाइन। स्टूडेंट्स, आज हम बायो क्लास 11 चैप्टर नंबर 12 एक्सरसाइज लॉन्ग क्वेश्चन आंसर सॉल्व करेंगे। सो लेट्स स्टार्ट। फर्स्ट क्वेश्चन हमारे पास, एक्सप्लेन मेंडल लॉ ऑफ इनहेरिटेंस ऑफ मोनोहाइब्रिड क्रॉस एंड इंटरप्रेट द रिजल्ट। मेंडल्स लॉ ऑफ मोनोहाइब्रिड क्रॉस, Gregor Mendel's law of inheritance, explain how traits are passed from one generation to the next. A monohybrid cross involves crossing two parents that differ in one trait. Mendel's monohybrid cross experiment. Mendel's crossed pure breed pea plant with contrasting trait parental generation tall into short. Mendel's hypothesis, law of segregation and law of independent assortment. According to law of segregation, each pair of allele separate during gamete formation, and according to law of independent assortment, alleles for different traits sort independently. Monohybrid cross result: first generation all offspring tall, second generation 3 by 4 tall and 1 by 4 short. Interpretation: dominant are recessive alleles, tall dominates short. Genotype and phenotype: F1 has tall phenotype but carries recessive allele. Segregation: alleles separate during gamete formation. And independent assortment: monohybrid cross demonstrates independent assortment. Monohybrid cross ratio: F1 100% tall, F2 3:1 tall/short. Key points: Mendel law applied to single gene trait. Monohybrid cross demonstrates segregation and independent assortment. Dominant and recessive alleles determine phenotype. Applications: predicting trait inheritance, understanding genetic diversity, and plant and animal breeding programs. Next question is, describe law of independent assortment under the following heads. A. Limitations, law of independent assortment, and usefulness, scope of law of independent assortment. Law of independent assortment. The law of independent assortment formulated by Gregor Mendel states that alleles for different genes are sorted independently of each other during gamete formation. Limitations: linked genes. Genes located close together on same chromosome tend to be inherited together. Genetic linkage. Linked genes violate independent assortment. Crossing over. Exchange of genetic material between homologous chromosome. Epistasis. Interaction between genes affect phenotype. Polygenic inheritance. Multiple genes induce a single trait. Gene clustering. Genes in close proximity on a chromosome. Next is usefulness and scope of the law of independent assortment. Predicting trait inheritance, help predict probabilities of trait combination. Genetic mapping, useful in constructing genetic map. Gene localization, identify gene location on chromosome. Breeding program, inform plant and animal breeding strategies. Genetic counseling, help in predicting genetic disorders. Forensic genetic, help in DNA profiling and identification. Evolutionary studies, explain genetic variation and diversity. Next is additional key points. Independent assortment occur during meiosis 1, applies to non-linked genes and increases genetic variation. Exceptions and modification: chromosomal linkage, genetic hitchhiking, and epistatic interaction. Modern applications: genome-wide association study, GWAS, personalized medicine, synthetic biology, genetic engineering. The law of independent assortment remains a fundamental concept in genetics, providing valuable insight into inheritance pattern and genetic diversity. Next question is, what is probability? How can you evaluate that inheritance of genes and their mixing during fertilization is based on mathematical probabilities? Probability is a measure of the likelihood of an event occurring ranging from 0, impossible, to 1, certain. Probability in genetics. Genetics uses probability to predict the inheritance of traits and gene combination. Key concepts: Mendel's law, segregation, independent assortment, and dominance. Punnett scale visual representation of probability calculation. Probability rules, multiplication rule, and addition rule. Evaluating probability in genetics. Monohybrid cross, probability of offspring genotype and phenotype. Example: tall 3 by 4, short 1 by 4. And dihybrid cross, probability of offspring genotype and phenotype. Example: tall and green 9 by 16. पॉलीजेनिक ट्रेट्स, प्रोबेबिलिटी ऑफ ट्रेट एक्सप्रेशन। पुनर्स के एग्जांपल, अगर हम क्रॉस कराएं दो हेट्रोजाइगस प्लांट्स को तो हमारे पास क्या आएगी प्रोबेबिलिटी ऑफ होमोजाइगस टाल 1/4, हेट्रोजाइगस टाल 1/2 एंड होमोजाइगस शॉर्ट 1/4। मैथमेटिकल प्रोबेबिलिटी फार्मूला, probability of genotype is equal to number of favorable outcome divided by total possible outcomes। and probability of phenotype कैसे कैलकुलेट करते हैं हम, probability of genotype 1 plus probability of genotype 2 plus so on। ठीक है, नेक्स्ट है इंपॉर्टेंस, predicting trait inheritance, understanding genetic variation, genetic counseling, breeding programs। Real world applications include genetic testing, personalized medicine, forensic genetic and evolutionary studies। Software and tools, Punerscare, genetic simulation software like Mendel's Law Simulator, and statistical genetic packages like or Python libraries। Next question is explain incomplete dominance with reference to the inheritance of flower in 4 o'clock plant। Incomplete dominance is phenomena where one allele does not completely mask effect of other allele, resulting in a blended or intermediate phenotype। 4 o'clock plant, Mirabilis dilapa, flower color red, dominate, white recessive, genotypes and phenotypes, R are for red and capital R small r for pink and small r for white। Punnett scale, when two heterozygous plants were crossed, we get a probability of homozygous red 1/4 and pink 1/2 and white 1/4. Incomplete dominance explanation. Red does not completely dominate white. RR genotype produce pink and intermediate phenotype. Neither allele is completely recessive or dominant. Key points. Incomplete dominance differs from complete dominance. Blended or intermediate phenotype. Both alleles contribute to the phenotype. Examples are snapdragon, carnations, and roses. Importance: understanding genetic variation, predicting trait inheritance, plant breeding programs, molecular basis, allelic interaction, gene expression regulation, protein-protein interactions. Next question is give a detailed account of ABO blood group system. A. Associate the positive and negative blood group with the presence and absence of Rh factor. B. Explain erythroblastosis fetalis in the light of antigen-antibody reaction. First one is ABO blood group system. The ABO blood group system is classification system for human blood based on the presence or absence of specific antigens or red blood cells. Antigens and antibodies. Antigens A and B substances on red blood cell. Antibodies A and B in plasma. ब्लड ग्रुप्स, अगर ब्लड ग्रुप ए है तो उसमें एंटीजन ए प्रेजेंट होगा और एंटीबॉडी बी प्रोड्यूस होगी प्लाज्मा में। एंड अगर ब्लड ग्रुप बी है तो एंटीजन बी होगा और एंटीबॉडी ए होगी। ए बी ब्लड ग्रुप है तो बोथ ए एंड बी एंटीजन होंगे, नो एंटीबॉडी होगी। अगर ब्लड ग्रुप ओ है तो नो एंटीजन होगा और बोथ एंटीबॉडीज ए एंड बी प्रेजेंट। आर एच फैक्टर अगर पॉजिटिव है तो आर एच डी एंटीजन प्रेजेंट होगा, अगर आर एच नेगेटिव है तो एब्सेंस ऑफ आर एच एंटीजन होगा। एसोसिएटिंग पॉजिटिव एंड नेगेटिव ब्लड ग्रुप विद आर एच फैक्टर, आर एच पॉजिटिव ए एंटीजन प्लस आर एच एंटीजन भी प्रेजेंट है। अगर बी पॉजिटिव किसी का ब्लड ग्रुप है तो उसमें बी एंटीजन और आर एच एंटीजन प्रेजेंट होगा। ए बी पॉजिटिव है, ए एंड बी एंटीजन और आर एच एंटीजन भी होगा। ओ पॉजिटिव है, नो ए बी और आर एच एंटीजन। सिमिलरली आर एच नेगेटिव की अगर हम बात करें तो ए नेगेटिव है तो उसमें ए एंटीजन बट नो आर एच एंटीजन। अगर बी नेगेटिव किसी का ब्लड ग्रुप है तो उसमें बी एंटीजन तो होगा लेकिन नो आर एच एंटीजन होगा। ए बी नेगेटिव, ए एंड बी एंटीजन प्रेजेंट बट नो आर एच एंटीजन। ओ नेगेटिव, नो ए बी आर, नो आर एच एंटीजन। नेक्स्ट इज erythroblastosis fetalis is a condition where maternal antibodies attack fetal red blood cells. Antigen antibody reaction, Rh negative mother carries anti-Rh antibodies. Rh positive fetus inherits Rh antigen from father. Maternal anti-Rh antibodies attack fetal red blood cells. Fetal red blood cells are destroyed, leading to anemia and potentially fatal outcomes. Prevention and treatment. Rogam anti-Rh immunoglobulin injection for Rh negative mothers. Screening for Rh incompatibility. Key points. ABO blood group system classify blood based on A and B antigen. Rh factor determine positive or negative blood group. Erythroblastosis fetalis occur due to antigen-antibody reaction. Next question is suggest prevention and treatment of erythroblastosis fetalis. It is a hemolytic disease of newborn caused by Rh incompatibility between mother and fetus. Prevention: Rh typing and screening, administering Rh immunoglobulin to Rh negative, and matching blood type for transfusions, genetic counseling. Treatment: prenatal monitoring fetal well-being, intrauterine transfusions IUT for severe EF, and postnatal exchange transfusion to remove Rh positive red blood cells, phototherapy to reduce bilirubin levels, supportive care, hydration, oxygenation. Newborn screening, bilirubin level testing, complete blood count, CBC, retics count. Complications management, Canistorus, bilirubin-induced brain damage, aggressive treatment, anemia, blood transfusion. Emerging research, non-invasive prenatal testing for EF prediction. Key organizations, American College of Obstetricians and Gynecologists, ACOG, American Academy of Pediatrics, AAP. Next question is explain polygenic inheritance with reference to wheat grain color. Polygenic inheritance refers to control of trait by multiple genes. और जो wheat का grain color होता है इसको भी multiple genes control कर रहे होते हैं. Wheat grain color is determined by multiple genes interacting to produce various shades of red, white or amber. Key genes are: 1. Controls production of red pigment. R2. Influence intensity of red color. W1. Suppress red pigment production. And A1 contributes to amber color. Polygenic interaction. R2 ये देखें अगर कैपिटल सब है तो ये डीप रेड होगा एक कैपिटल अगर और एक स्मॉल है हेटेरोजाइगस कंडीशन है तो मीडियम रेड होगा और रिसेसिव है तो वाइट एम्बर कलर होगा पॉलीजेनिक इनहेरिटेंस प्रिंसिपल्स multiple genes contribute to the trait. Each gene has a small effect. Gene interactions determine the final phenotype. Continuous variation in the trait. Characteristics: quantitative trait locus, QTL mapping, additive and non-additive gene interaction, environmental influences. Other examples: human height and skin color, plant trait like yield, disease, resistance, animal coat color, and growth rate. Importance: understanding complex trait, improving crop yield and disease resistance for breeding programs. Molecular techniques, QTL mapping, genome-wide association study, GWAS, gene editing, key points. Multiple gene control with grain color, polygenic inheritance, explain continuous variation, gene interactions determine trait expression. Next question is what is epistasis? Explain relationship of epistasis with polygenic inheritance. Epistasis is the interaction between genes of different loci that affect the expression of trait, masking or modifying the effect of one gene by another. Types of epistasis: dominant epistasis, one gene mask other; recessive epistasis, one gene reveals another; additive epistasis, gene combine effect; and multiplicative epistasis, gene interactions multiply. Relationship with polygenic inheritance. Epistasis play a crucial role in polygenic inheritance. Gene-gene interaction. Epistasis influence trait expression, masking and modifying effect. Epistasis alter gene contribution and complex trait architecture. Epistasis contribute to continuous variation. Polygenic epistasis models: additive model gene and effect, multiplicative model gene interactions multiply, threshold model epistasis determine trait expression. Examples are wheat grain color, human height, plant disease resistant gene for gene interactions. Impact on trait expression: reduced or increased trait expression, altered trait distribution, and increased genetic variation. Detection methods: quantitative trait locus QTL mapping, GWAS, and gene-gene interaction analysis. Software tools: epistasis detection software Epistase and genetic analysis software R or Q2L. Key points: epistasis influence polygenic inheritance, gene-gene interactions modify trait expression, epistasis contribute to complex trait architecture. Next question is explain epistasis with reference to the inheritance of coat color in Labrador Retriever. Labrador coat color is determined by multiple genes interacting through epistasis. Key genes कौन कौन से हैं इसमें? Capital B for black, which code for black pigment. Small b, which code for brown pigment. Capital E, control production of eumelanin, black brown pigment, and small e reduces eumelanin production. Epistasis interaction क्या है? capital B या capital B small b, black plus E, black coat. B capital B small e dark brown. Small b capital E small e brown, and capital B small e light brown cat coat. Epistatic effects. Dominant epistasis, capital E gene mask, small b gene. Recessive epistasis, small e genotype reveal B gene. And additive epistasis, genotype produce darker black coat. कोट कलर जीनोटाइप एंड फिनोटाइप, ये देखिए जीनोटाइप भी दी हुई है और इसकी फिनोटाइप भी है। अगर सारे जो है ना डोमिनेंट है तो उसका कलर क्या होगा? ब्लैक। अगर जो है हमारे पास कैपिटल ई स्मॉल ई है तो भी ब्लैक होगा। अगर दोनों रिसेसिव है ई वाले तो डार्क ब्राउन। अगर बी वाला क्या है हेट्रोजाइगस तब भी ब्लैक। अगर दोनों हेट्रोजाइगस है तो डार्क ब्राउन। अगर बी वाला जो हमारे पास हेट्रोजाइगस है जबकि ई वाला रिसेसिव है तो ब्राउन। बी वाला अगर रिसेसिव है और ई वाला डोमिनेंट है तो ब्राउन। बी वाला रिसेसिव और ये हेट्रोजाइगस तो लाइट ब्राउन, और अगर और रिसेसिव है तो लाइट ब्राउन। Implications. Epistasis influence coat color variation. Multiple genes contribute to complex trait. Gene interaction determine phenotype. Breeding programs. Predicting coat color probabilities. Selecting breeding pairs for desired traits. Key points. Epistasis control Labrador Retriever coat color. Multiple genes interact to produce various phenotype. Understanding epistasis inform breeding programs. Next question is, explain epistasis with reference to inheritance of flower color, pigment phenotype of sweet pea Lethyrus odoratus. Sweet pea flower color is a classic example of epistasis demonstrating gene-gene interaction controlling pigment phenotype. Genes involved are capital R for red genes, P for purple and W for white. Epistatic interactions. देखिए अगर ऑल डोमिनेंट है तो हमारे पास पर्पल कलर है। अगर आर और पी जो जीन्स हैं वो हेट्रोजाइगस है तो रेड पर्पल। अगर आर वाला रिसेसिव है तो वाइट पर्पल। पी वाला रिसेसिव है तो रेड। आर वाला जो है हमारे पास हेट्रोजाइगस और पी रिसेसिव है तो पिंक। एंड रेड और पिंक वाले दोनों ही रिसेसिव है तो वाइट। एपिस्टेसिस टाइप्स। डोमिनेंट एपिस्टेसिस आर जीन जो है ना उसने मास्क कर दिया था पी जीन को, ठीक है। इसी तरह से रिसेसिव एपिस्टेसिस हुई है जो स्मॉल रिसेसिव आर जीनोटाइप है उसने रिवील किया था डब्ल्यू जीन को। एंड एडिटिव एपिस्टेसिस कैसे हुई है? पी combination produce red purple color. Key findings. Multiple genes interact to determine flower color. Epistasis modify gene effect. Gene combination produce novel phenotypes. Mendelian genetic. Inheritors follow Mendelian law. Gene interaction illustrate epistasis. Importance. Understanding complex trait demonstrating epistasis in plant breeding. Insights into genetic control of pigment production. Other examples: Petunia, flower color, corn kernel color, and human skin pigmentation. Molecular basis: gene regulation, expression, protein-protein interaction, metabolic pathway interaction. Key points: sweet pea flower color exhibit epistasis, gene-gene interaction control pigment phenotype, epistasis contribute to complex trait architecture. Next question is, what is gene linkage? How gene linkage can be detected? Explain Morgan's experiment. Suggest why linkage could be observed or evaluated if the number of progeny is quite large. Gene linkage. It refers to the physical proximity of genes on a chromosome leading to their joint inheritance. Gene linkage characteristics. Linked genes tend to be inherited together. Frequency of recombination between linked genes is reduced. Linked genes can be mapped to specific chromosomal region. Detection method. Crossing over frequency analysis, linkage mapping, genetic linkage analysis, physical mapping. Morgan's experiment in 1910. Thomas Hunt Morgan Drosophila melanogaster fruit fly experiment demonstrated जीन लिंकेज। अच्छा 1910 में थॉमस एंड मॉर्गन ने क्या किया था, जो फ्रूट फ्लाई होती है जिसका नाम होता है हमारे पास साइंटिफिक नेम ड्रोसोफिला melanogaster, उसके ऊपर उसने एक्सपेरिमेंट परफॉर्म किया था जिससे जीन लिंकेज हमें उसने शो किया था। एक्सपेरिमेंटल डिजाइन पेरेंटल जनरेशन थी येलो एंड वाइट आइड फ्लाइज। वन जनरेशन जो आई हमारे पास क्रॉस बिटवीन येलो एंड वाइट आइड फ्लाइज एंड F2 जनरेशन इंटर F1 offspring. Results yellow and white eye colors did not segregate independently. More yellow-white combination observed than expected. Conclusion: linked genes Y and W on X chromosome reduce recombination frequency. While linkage can be observed evaluated with large progeny, increased statistical power, reduced sampling error, more accurate estimation of recombination frequencies, detection of subtle linkage effects. Large progeny advantages: identification of closely linked genes, construction of high-resolution genetic map, improved precision in gene localization. Importance: understanding gene organization, mapping disease genes, farming breeding programs. Key points: gene linkage affect inheritance pattern. Morgan's experiment demonstrated linkage. Large progeny sizes enhances linkage detection. Next question is: explain Morgan's experiment to demonstrate the role of crossing over in gene linkage. Morgan's experiment 1910 demonstrating crossing over in gene linkage. Thomas and Morgan Drosophila melanogaster fruit fly experiment showcase the role of crossing over in gene linkage. Experimental design. Parental generation yellow-eyed and white-eyed and normal-eyed flies. X chromosome and F1 generation cross between yellow-white-eyed and normal-eyed male XY. F2 generation interbred F1 offspring. Goals: investigate inheritance of yellow and white eye color, understand gene linkage and crossing over. Results: F1 offspring, all female flies had yellow eye while males have normal eyes. F2 generation, 95.5% of flies exhibit linked traits, yellow-white, 4.5% showed crossing over. Conclusion: linked genes YW on the X chromosome, crossing over occur between linked genes. Frequency of crossing over inversely relate to the gene distance. Key findings: crossing over frequency 4.5% later refined to 1.3 map units. Gene leakage demonstrated. Chiasma formation during meiosis facilitate crossing over. Significance: confirmed Sutton-Boveri hypothesis. Chromosomes carry genetic information. Established crossing over as a fundamental genetic process. Laid foundation for modern genetics and genomics. Morgan's contribution: first genetic map 1911 demonstrated chromosome theory of inheritance and paved the way for genetic linkage analysis. Key points: Morgan experiment demonstrated gene linkage. Crossing over occurs between linked genes. Frequency of crossing over relates to gene distance. Next question is explain genetic identification of phenotypes in Drosophila in man. Drosophila and man. Genetic identification in Drosophila fruit fly. XY chromosome system XX जो थे वो फीमेल एंड XY मेल ऑटोसोमल जीन्स डिटेक्ट की जीन्स TRA DSXL SXL genetic mechanism X chromosome it activate SXL and Y chromosome no activation in and in case of human XY क्रोमोसोम सिस्टम होता है एक्स एक्स फीमेल एंड एक्स वाई मेल ऑटोसोमल जीन इन्फ्लुएंस्ड डेवलपमेंट की जीन्स कौन से होते हैं SRY एंड SOX9 जीन SRY बॉक्स 9 and DX1 dosage sensitive reversal genetic mechanism Y chromosome SRY expression and X chromosome absence of SRY comparison Similar XY chromosome system, different key genes and mechanism, conservation of genetic principles, and diagnostic techniques, karyotyping, PCR, polymerization reaction, genetic sequencing. Next is explain sex linkage in Drosophila. It refers to the inheritance of gene located on X and Y chromosome. X chromosome female and Y chromosome male specific. Genes are X-linked genes which are located on X chromosome and Y-linked genes which are located on Y chromosome. Inheritance pattern. X-linked recessive traits. One copy of allele sufficient for expression. And single XY single allele determines trait. X-linked dominant traits. One copy of allele sufficient for expression and XY single allele determined trait. Examples are आई कलर रेड जो होता है वो वाइल्ड टाइप है, जो व्हाइट है वो म्यूटेंट है. मॉर्गन एक्सपेरिमेंट इन 1910. एक्सपेरिमेंटल डिजाइन क्या था? पेरेंटल जनरेशन व्हाइट आइड फ्लाइज को क्रॉस करवाया था रेड आइड फ्लाइज के साथ. जनरेशन ऑल रेड आइज एंड ऑल मेज हैव व्हाइट आइज. कंक्लूजन. आई कलर जीन लोकेटेड ऑन एक्स क्रोमोसोम. नेक्स्ट क्वेश्चन इज explain sex linkage in man. It refers to the inheritance of traits carried on chromosome X and Y. Characteristics: gene located on X chromosome and two copies of the gene, and a male XY one copy of the gene. Patterns: X-linked recessive and X-linked dominant. Males more frequently affected, females more affected. X-linked dominant जो होगा वो females more frequently affected. Examples: hemophilia A, red green color blindness, vitamin D resistant rickets. Y-linked inheritance characteristics: genes located on Y chromosome, XY one copy of the gene and exists no Y-linked genes. Patterns: Y-linked traits exclusive to males and Y-linked disorders rare. Examples: inheritance risk, genetic counseling, genetic testing, and risk assessment. Next question is explain genetics of hemophilia. Hemophilia is genetic disorder characterized by impaired blood clotting. Types: दो टाइप्स हैं हीमोफीलिया की, हीमोफीलिया ए, हीमोफीलिया बी, फैक्टर ए डेफिशिएंसी एंड फैक्टर 9 डेफिशिएंसी. इनहेरिटेंस पैटर्न एक्स लिंक्ड रिसेसिव ट्रेट है. फैक्ट्री जीन्स F8 हीमोफीलिया ए, F9 हीमोफीलिया बी एंड एक्स क्रोमोसोम. जेनेटिक मैकेनिज्म की बात करें तो म्यूटेशन इन F8 और F9 जीन disrupt clotting factor production. Normal gene code for functional clotting factor. Mutant gene lead to non-functional or reduced clotting factor. Expression male XY. Single copy of mutant gene causes hemophilia. Female two copy of mutant gene cause hemophilia. One copy makes them carrier. Then आप ये देख सकते हैं symptoms: prolonged bleeding, joint pain and swelling, muscle weakness. Diagnosis: genetic testing and blood clotting assay. Treatment: clotting factor replacement therapy या gene therapy. Key points: hemophilia is an X-linked recessive disorder. Mutations in F8 or F9 gene cause hemophilia. And a famous example: Queen Victoria descendants, for example Prince Leopold, Prince Lexi of Russia, suffered from hemophilia. Next question is explain genetic of color blindness. Color blindness, also known as color vision deficiency, is a condition where individuals have difficulty perceiving certain colors. Genetic basis: X-linked recessive inheritance pattern, mutations in core cell genes and located on X chromosome. Key genes are L-cone opsin code for long wavelength cone pigment and M-cone opsin which code for medium wavelength cone pigment green. Color blindness types: थ्री टाइप्स होती हैं. रेड ग्रीन कलर ब्लाइंडनेस मोस्ट कॉमन होती है ये. प्रोटानोपिया रेड ब्लाइंडनेस. म्यूटेशंस इन ओपीएन 1 LW एंड ड्यूटरानोपिया ग्रीन ब्लाइंडनेस जिसको हम कहते हैं, इसमें म्यूटेशन होती है ओपीएन 1 MW में. टोटल कलर ब्लाइंडनेस होती है जो रेयर होती है, जिसको हम कहते हैं एक्रोमेटोप्सिया. इनहेरिटेंस पैटर्न क्या होता है ये देख लें आप. डायग्नोसिस हम इनको इन 3 टेस्ट के थ्रू कर सकते हैं: इजायरा टेस्ट, जेनेटिक टेस्टिंग एंड ट्रीटमेंट. नो क्योर बट color correction glasses and technology assistance can help. Key points क्या हैं? Color blindness is excellent recessive mutations in these genes and red green color blindness most common होती है. Next question is explain genetics of muscular dystrophy. Muscular dystrophy (MD) is group of genetic disorder characterized by progressive muscle weakness and degeneration. अब इसकी भी टाइप्स हैं. टाइप्स ऑफ मसल डिस्ट्रॉफी ये आर Duchenne muscle dystrophy DMD, Becker muscular dystrophy BMD, limb girdle muscular dystrophy LGMD, facioscapulohumeral muscular dystrophy FSGD, myotonic dystrophy DM. जेनेटिक काजेज की बात करें तो म्यूटेशंस इन डिस्ट्रोफिन जीन में हो जाती हैं, म्यूटेशंस इन सार्कोग्लाइकन जीन्स एंड म्यूटेशंस इन FSHD and expansion of CTG repeats in myotonic dystrophy gene. Dystrophin gene. It is located on X chromosome, code for dystrophin protein essential for muscle function. Inheritance pattern. It is X-linked recessive and ये देख लिया autosomal recessive and autosomal dominant. Genetic testing, DNA sequencing, PCR, polymerase chain action, genetic linkage analysis, symptoms and diagnosis की बात करें, progressive muscle weakness, muscle wasting, delayed motor development, elevated creatine kinase level. Treatment and management, physical therapy, occupational therapy, speech therapy, medication, and gene therapy. Next is key points. Muscular dystrophy genetic disorder है, multiple types with distinct genetic causes, X-linked recessive inheritance common, genetic testing essential for diagnosis, management and treatment focus on symptom relief. Next is describe sex-related traits. Characteristics influenced by individual chromosomes X and Y and their primary and secondary traits. प्राइमरी ट्रेड्स में आप ये देख सकते हैं and secondary traits then, and these traits include height, weight, hair loss, etc. Linked traits include hemophilia A and B, color blindness, Duchenne muscular dystrophy. Inheritance patterns की अगर हम बात करें तो ऑटोसोमल हैं, X linked located on X chromosome and Y linked भी होते हैं which are present on Y chromosome. जेनेटिक एंड एनवायरमेंटल फैक्टर्स में हमारे पास आ जाते हैं जीन एक्सप्रेशन, हार्मोनल बैलेंस, एनवायरमेंटल इन्फ्लुएंस लाइक डाइट एंड लाइफस्टाइल, क्लिनिकल सिग्निफिकेंस, अंडरस्टैंडिंग डेवलपमेंट डिसऑर्डर एंड डायग्नोस्टिक जेनेटिक डिसऑर्डर. की पॉइंट्स, these are influenced by chromosomes X and Y, inheritance pattern and hormonal balance play crucial role. So students, हमारे ये लॉन्ग क्वेश्चन भी कंप्लीट हो चुके हैं. होपफुली आपको समझ आ गए होंगे. इफ यू हैव एनी डाउट यू कैन आस्क इन कमेंट सेक्शन. प्लीज लाइक शेयर एंड सब्सक्राइब माय यूट्यूब चैनल. थैंक यू.